All right. Thank you so much for that kind introduction. And first, I would like to extend my thanks to the ICP committee for giving us this opportunity to present our work at this webinar. And so let's get going. So to start motivating this presentation, as you might be aware, cerebrovascular disease is a major risk factor for dementia due to Alzheimer's disease. And therefore, if we want to solve the current Alzheimer's disease crisis, it's very important that we understand the interactions between cerebrovascular disease and Alzheimer's disease. One of the major limitations to study these AD and vascular interactions is the lack of sensitive and specific markers of cerebrovascular health. For example, traditionally, vascular health is characterized using systemic markers such as blood pressure, cholesterol levels, and genetics. And if we want to probe vascular disease in the brain, patients typically undergo an MRI that assesses structural markers such as white matter hyperintensities, microbleeds, and diffusion. However, in the majority of these markers only indirectly probe cerebrovascular disease and are more sensitive to downstream effects of vascular changes. With recent advances in quantitative MRI, functional vascular imaging is now possible, including flow imaging. But it's also very important to notice the diversity of the vascular landscape in the brain. This includes the micro and the micro circulation, and it's this diversity that drives technology development. And ideally, to truly understand CVD and AD interactions, we need to probe each one of these compartments. In using phase contrast MRI, more specifically 4D-Flow MRI, we can directly probe the cerebral arteries and veins and indirectly the capillary bed. So traditionally, 4D-Flow MRI clinical implementations have been burdened by long scan times. However, today there are many different approaches that we can use to allow for clinically feasible scan times. For example, one way to accelerate the acquisition is during the data acquisition step, particularly. And in this example here, what we're showing is the PC VIPR principle that exploits the fact that the bulk image contrast information is located in the center of case space. And therefore, using a radial sampling trajectory, we can heavily oversample the center of case space, but save imaging time by undersampling the higher frequencies. And this leads to accurate images with 10 to 30 times less data, given that the data are sparse or compressible. So using this approach, we can acquire 4D-Flow MRI scans at 3 Tesla with whole brain coverage, 0.7 millimeter isotropic spatial resolution, and roughly 50 milliseconds temporal resolution, all this in 6 minutes. And this type of acquisition is robust to motion artifacts because of the short echo times, and also the undersampling artifacts are relatively benign. And here you can see an example of velocity streamlines along the circular Willis. So with 4D-Flow, we have the opportunity to measure intracranial pulse wave velocity, which is a biomarker for arterial stiffness, overcoming the limitations of other traditional methods, such as ablanation tonometry and transcranial Doppler ultrasound. However, intracranial pulse wave velocity measures require high temporal resolution. And in order to increase temporal resolution, we can reconstruct more time frames, but by further undersampling our data, we also increase the noise in our images. To alleviate these problems, we can use constrained image reconstruction methods, such as a local low rank, which have been shown to denoise highly undersampled data sets. And we can further push the theoretical temporal resolution limit by splitting the flow encodes during the reconstruction step. And what I mean with that is here's a cartoonish representation of how this reconstruction combined with flow encode splitting will help generate artifact-free images. So here we have a standard 4D-Flow recon. We usually acquire 4 measurements to encode velocities in X, Y, and Z, and 1 measurement to remove the background phase. And in this example, we are using a 4-point reference velocity encoding scheme. And here we have a cardiac waveform that we want to reconstruct. So we start dividing the cardiac cycle into multiple beans where data will be shared on average. And then we define a timestamp long enough to include all 4 encodes. And this is used to assign projections to beans during the reconstruction. This enforces that the data with the same projection angle is added to the beams, which also sets our theoretical temporal resolution limit to 4 TRs. Then after enough data has been collected, we have a relatively uniform sample case space for each of the encodes. Then we perform gradient and sense reconstructions, and we obtain an image for each of the encodes, where the reference image is subtracted from the velocity data to remove the background field offsets. However, if we shorten this timestamp to split the encodes and has a theoretical higher temporal resolution, for example, in this case, we split them into 2 encodes, the resulting overall coverage of case space for each encode pair will be different. And then after reconstruction and image subtraction, the different projection angles on each encode pair will lead to different artifacts, which can end up as residual artifacts in the velocity data. Instead, if we use the local low rank approach, information from all the beams is used during the reconstruction, and the missing projections are recovered to a certain extent, ultimately helping reduce potential for residual artifacts. And we tested this approach in simulations where we combine the local low rank with the flow encode split. And here you can see an example where we have 2 phantoms. Each one has 2 vessels. The phantoms were generated using opposite velocity encodings or banks. And these phantoms were radially sampled using a golden angle distribution and 1,000 projections on a 2D grid. Then the sample data, raw data, was reconstructed using a sense and a sense combined with local low rank reconstruction with and without encode split. After the reconstruction, we do complex division between the encodes to generate a resulting time series. And this is the results that we get. We can appreciate that the local low rank reconstruction is less sensitive to undersampling and residual artifacts compared to the sense reconstruction with and without encode split. So using these methods, we were able to quantify intracranial pulse wave velocity using 4D-Flow in a population of clinically diagnosed Alzheimer's disease patients. And what we found was that pulse wave velocity was significantly higher in the AD patients when compared to age-matched controls. But very interestingly, if you look at the plot on the far right, there are 2 groups that they are middle-aged adults, they're younger, they're cognitively healthy. The only difference between these 2 groups is that one is at lower risk of dementia due to AD and the other one is at higher risk. And the group at high risk, the far right, what I mean is that they have parental history of dementia due to Alzheimer's disease, and they are also ApoE epsilon 4 positive. And this group of normal subjects is showing already higher pulse wave velocity compared to the one at lower risk, suggesting that vascular alterations are happening. More people are still cognitively normal. In our healthy populations, at lower risk, we observe that pulse wave velocity increases with age, and while pulse wave velocity suggests arterial stiffening in the Alzheimer's disease population, and is an effect that appears to show to accelerate the aging effect on these AD populations. So that study was limited to subjects with AD clinical syndrome and did not characterize cerebrovascular health during the preclinical phase of Alzheimer's disease, which begins with the accumulation of amyloid pathology in the brain, and it can last decades. And it's during this phase that pathophysiological changes are happening, and it's likely when therapeutic interventions will be most effective. So in our follow-up study, we characterized intracranial vascular health in 80 biomarker confirmed subjects, and we also included traditional markers of CVD, such as white matter hyperintensities, microhemorrhages, perivascular spaces, and 80 biomarkers were determined from PET imaging, including PIP and MK6240, from which we quantify amyloid and tau burden. And a total of 136 subjects were recruited from the Wisconsin Alzheimer's Disease Research Center and the Wisconsin Registry for Alzheimer Prevention. 4 groups were established based on cognitive status, amyloid, and tau burden. And please notice that 3 out of the 4 groups were cognitively unimpaired. And our findings show that traditional CVD markers, including blood pressure, heart rate, white matter hyperintensities, and perivascular spaces, were similar between these groups. However, we noticed the significant differences in the presence of microhemorrhages between age-matched controls and amyloid and tau biomarker positive subjects that were cognitively normal, indicating perhaps the presence of cerebral amyloid angiopathy or a small vessel disease. when we look at the measurements of total cerebral brain blood flow, it was independent of AD biomarker status after we adjusted for neuron coupled metabolism by brain volume normalization. And one thing that we can measure with 4D-Flow is the dynamic evolution of the cardiac pulse wave along the vasculature. For example, the pumping action of the heart generates a pulse wave, and this pulse wave travels through the intracranial arteries, reaching the capillary bed, and then it returns to the heart through the venous system. And by characterizing the cardiac pulse wave as it travels from the intracranial arteries to the brain, parenchyma, and finally to the veins, we can establish the trans-capillary pulse wave delay transit times. And these transit times are indicators of pulse wave transmission and vascular compliance of the macro and micro circulation. And here you can see a summary of the trans-capillary pulse wave delay measurements for the 4 groups measuring the internal carotid artery and the basilar artery. And in our study, we found that significantly shorter transit times were associated with faster pulse wave transmissions in AD biomarker positive subjects. And these changes were also detected in cognitively normal subjects, that is during the preclinical phase of AD. And these results are indicative of vessel and or capillary stiffness. With 4D-Flow MRI, we can also study bit-to-bit blood flow fluctuations. And in this example, you can see blood flow changes in the superoccipital sinus, which is a large intracranial draining vessel. And from these fluctuations, we can derive low-frequency flow oscillations. And please notice that instead of doing cardiac resolve reconstruction, now we're doing time resolve reconstruction with temporal resolution of the order of 3.4 seconds. And since I hit the 12-minute mark, I'm going to skip to the interesting results and to the discussion. And from those time resolved images, we estimated the low frequency oscillation content from power spectral density analysis in the arteries and veins. And in these measurements, we also find a significant reduction of arterial low frequency oscillation content in AT biomarker positive subjects, indicating a reduction of perhaps arterial vasomotion driven, smooth muscle cell driven vasomotion. Again, during the preclinical stage of AD. And hopefully I have conveyed that we can look at high frequency cardiac pulsation, but also at these lower frequency oscillations, different components of the vascular system, and that we can go beyond traditional and structural markers of cerebrovascular disease to more specific markers of vascular health of relevance to Alzheimer's disease. These results suggest that perhaps there is a synergistic interaction between Alzheimer's disease and these vascular alterations, and they are happening during the preclinical stage of Alzheimer's disease. However, we still need longitudinal studies to elucidate the interaction pathways. And finally, these vascular alterations might help explain the observed heterogeneity that we see in the neuropathologic trajectory of Alzheimer's patients. And sorry for going a little bit over the time, but I'd like to thank you for your attention. Take any questions you might have. We still have a minute and a half. Thank you.